Thank you for joining us for today's webinar and discussion on begomoviruses and cucurbits, hosted by the Emerging Viruses and Cucurbits Working Group. I'm Rebecca Melanson, an extension plant pathologist with the Mississippi State University Extension Service, co-chair of the EVCWG, and co-host for today's webinar and panel discussion.
And I'm Bill Wintermantel, a research plant pathologist and virologist with the USDA Agricultural Research Service in Salinas, California, also co-chair of the Emerging Viruses and Cucurbits Working Group, and your other co-host for today's webinar and panel discussion.
The Emerging Viruses and Cucurbits Working Group was established in March 2022 with the mission to improve communication and knowledge about viruses across the cucurbit industry and develop strategies to successfully identify and mitigate virus threats to cucurbit production in the United States. In the first 2 years, the Emerging Viruses and Cucurbits Working Group delivered presentations to various stakeholder groups, and developed several educational resources, including fact sheets, postcards, and videos on emerging viruses and cucurbits, the latter of which are freely available on the EVCWG website, which you can access by scanning the QR code on the screen, or by visiting ecucurbitviruses.org. We have since expanded our outreach activities to include opportunities such as webinars and open meetings for stakeholder input and participation in the emerging viruses and cucurbits discussion. The EVCWG and its outreach activities have been supported through competitive funding provided through 2 Southern IPM Center grants. Today's webinar is the second webinar we have offered. If you missed the first webinar on watermelon chlorotic stunt virus, we are in the process of getting this presentation posted to the website, and you will be able to access that presentation in the near future. If you would like to be notified of additional opportunities for participation or additional resources by the EVCWG, we encourage you to sign up for the EVCWG mailing list. You can do so by clicking the link provided that I will provide in the chat momentarily, or you can click the Join Our Mailing List button on the EVCWG website homepage at any time. While today's webinar and discussion are hosted by the EVCWG to provide information and stimulate discussion, any opinions, findings, conclusions, or recommendations expressed in the webinar and discussion are those of the authors and do not necessarily reflect the view of USDA, the Southern IPM Center, or the EVCWG. In a video of today's webinar, the presentation will be available on the EVCWG website in the near future. The video will only include the presentation and not the panel discussion.
Today's webinar presenter is Dr. Bob Gilbertson. Dr. Gilbertson is a distinguished professor of plant pathology in the Department of Plant Pathology at the University of California, Davis. His research has focused on pathogens transmitted through seed and on insect-vectored viruses of vegetable crops, including cucurbits. This has involved applied and basic research with an emphasis on developing effective IPM approaches. Bob's research group has published 184 papers in peer-reviewed journals, and this work has been recognized with numerous awards, including Fellow of AAAS and APS. He is an accomplished mentor and teacher at the undergraduate and graduate levels, and currently teaches an undergraduate course titled Feeding the World.
First, before we start, I'd like to thank Bill and Rebecca for the invitation to give this presentation. And initially, we were going to focus just on the seed aspect of this topic, but then we decided to expand it a little into cucurbit infecting begomoviruses in general. So what I plan to do is give you a very brief overview of Gemini viruses, because Bill's already talked about watermelon chlorotic stunt. He's introduced you to Gemini viruses in the previous webinar. So I won't go into a lot of details on that. We'll get right into the begomoviruses. And then I'm going to take you on a little journey, a fantastic voyage through the plant, okay? Where you're actually going to try to follow the virus all the way up into the flowers and seed of a plant. Now, this is going to be in common beans, not in cucurbits, but It's really a cool story, okay? And then we'll shift into cucurbit infecting begomoviruses and talk about them with the focus on cucurbit leaf crumple virus, which many of you know and has emerged as a significant problem in the southeastern U.S. Ironically, it has really, it first showed up in the southwestern U.S., but it's really not much of a problem there these days. It's really become more of a problem in Florida. And then, so we'll talk a little about how the plants actually defend themselves against this virus, and then we'll move into the discussion of seed and the seed association of begomoviruses, okay? And that will be like the third part of the presentation today. So I'll have to move quickly through this, but hopefully it will give you a good idea of these viruses, something about how they move around in plants, and I think it will convince you that these viruses are able to access the reproductive organs of the plant. And then, of course, the big question is, do they get into the seeds? And is this on the seeds or in the seeds? And of course, as this isn't a new question, we've had this question about other pathogens, bacterial canker notably. Is it in or on the seed? And those experiments are not easy to do. You need to have good numbers and you need to have good assays. And that's why the research techniques that are used in these studies are very important, okay? And we'll introduce you to the non-vector techniques of delivering these viruses to plants, okay? And when you think about that, you need to remember these are laboratory methods. So we'll mostly talk about agro-inoculation and particle bombardment inoculation. These are the two non-vector methods that are most commonly used because most of these viruses, like cucurbit leaf crumple, are not mechanically transmissible, okay? Unfortunately, some of them are, though. Some bipartite begomos are mechanically transmissible, but cucurbit leaf crumple is not one of them, at least in our hands. And it's a phloem-limited virus, so it is limited to the phloem, okay, which is consistent with the lack of mechanical transmission. Mechanically transmitted viruses tend to escape the phloem, and some Geminis do, not that they get that far away from it, but they can be mechanically transmitted, and therefore and you can show that they escape that phloem limitation. So there's a lot of good new information about cucurbit begomoviruses that I'm going to try to bring together and then take us through some of these examples of the association with the viruses with seeds. And we'll go through some of the techniques that were used and some of the conclusion results and see if we can come up with some consensus. But always remember, each one of these interactions, it's going to depend on the strain of the virus, the cultivar of the plant, and of course the species and the environment and all these other issues. We want to make definitive conclusions about things, but if you've worked on Gemini viruses as long as I have, one thing you learn is that they're paradigm breakers, okay? They have broken so many paradigms, it isn't even funny. Them being seed transmitted would hardly be earth-shattering in terms of knowing, just for example, we never thought they replicated in insects, but there's evidence now that TYLCD specifically replicates in the salivary glands of certain of the whitefly vectors, okay? And of course, there's a transovarial transmission that's been shown in TY that we didn't think would happen, okay? We didn't think there was indigenous monopartite begomoviruses in the New World. Now we know there's a whole bunch of indigenous monopartite ones. So it's not just old world monos and new world bipartites. Those paradigms have also been broken. This has just been, in my experience working with Gemini viruses, constant. How about aphid transmission? Who would have thought Geminis were aphid transmitted? The capuloviruses, that genus of Geminis are aphid transmitted. Breaking paradigms is something Gemini viruses and begomoviruses in particular have a habit of doing. So we don't want to get set in our opinions and everything, particularly about these viruses, because they are quite flexible. And another thing was, oh, they're single-stranded DNA viruses, so they're not going to have a high mutation rate. They have a mutation rate that's equal to plus-sense RNA viruses. So once again, another paradigm broken. People used to say, oh, they're not going to recombine. They're DNA viruses. These viruses recombine more than any virus, any RNA virus I know. Okay. So their ability to recombine is unbelievable. Good morning here in California and to wherever you all are. And thank you for joining this webinar. I'm quite excited about talking about one of my favorite topics, which is Gemini viruses, and in particular, cucurbit begomoviruses. Okay. I'm going to make an assumption that many of you know about viruses already. You already know that most of the families of plant viruses have genomes of plus-sense RNA, and it seems to work well for them, being able to translate their genomes immediately upon entering the cell. However, there are a number of groups of single-stranded DNA viruses, even though there's only two families of them, the nanoviridae and the geminiviridae, that are extremely important. And they have a large number of members, particularly the geminiviridae, which may in fact be the largest family of viruses that's even known now, okay? All right, so we're going to talk about that group of viruses, okay? And they're very interesting because they started out as an obscure group of viruses, and now, as I mentioned, they're one of the largest that we know today, okay? So what are Gemini viruses, okay? They're characterized by their characteristic virions, okay? So if you grind up infected plants and you're patient enough to try to purify them, you see these twin icosahedral particles. They look like two soccer balls stuck together. They're 18 by 30 nanometers. They're small and they're very unstable, okay? So purifying them is no easy task, okay? And they're not stable viruses, okay? And that's why they were hard to work with back in the day, okay? Which I have to say, I was right at the beginnings. All right, their genome size is small. It can be as little as one circular single-stranded DNA of 2.6 thousand nucleotides, 2.6 K. That's a very small genome. Some of them have a bipartite genome of two single-stranded DNAs called an A and a B. They're both about 2.6 KB, and their genome thus is 5.2 KB. In fact, those are the ones we're going to be talking about today, the bipartite ones, okay? They're transmitted by various insects. I told you, aphids, leafhoppers, whiteflies, etc., okay? And many cause devastating diseases like the tomato yellow leaf curl down here that we'll talk a little bit about today too, okay? All right, now I have to tell you a little about my personal story because I was a postdoc at the University of Wisconsin working on Xanthomonas, a bacteria of common bean that I liked a lot. And we were funded by USAID and to work in Brazil. And one day my boss, Doug Maxwell, I didn't put his face up here, but called me into the office and, Bob, they're cutting our funding in Brazil because Brazil graduated to a graduate country, so we can't work there anymore. So I'm sitting there going, oh boy. And then Doug says, but they said they'll continue the project if we'll work on bean golden mosaic virus. So I said, oh, okay. And of course, Doug, in his usual, well, Bob, do you want to work on bean golden mosaic virus? I said, yeah, okay. Of course, I want my job. By the way, what is bean golden mosaic virus? Okay. Now, I also tell my students in the graduate program at UC Davis, you need to be trained in everything because you never know what you'll work on. Of course, some of my colleagues don't agree with me. You should be trained in bacteriology, virology, and mycology. Okay? And in my PhD, I studied Fusarium. Then, as I said, I went and did a postdoc on Xanthomonas, and then I had to work on bean golden mosaic virus. Okay? You cannot just say when you get your PhD, oh, I'm going to work on Fusarium for the rest of my career. Obviously, that didn't happen in my case. There's bean golden mosaic on the lower left, okay? And it turned out when I started to work on Gemini viruses, there were 4 genera, okay? And this was in the late 1970s, early 1980s. In the late 1980s, sorry, early 1990s, okay? And so there were 4 genera. Mastery virus, which infects monocotyledonous plants, Curtovirus, and Begomovirus, and then this little Topoq virus, which is tomato pseudocurlytop, a curiosity disease only found in Florida and has never moved from there, okay? Those were the four when I started working, and I worked on bean golden mosaic, okay? But many of you are familiar with this technology called rolling circle amplification, where you can take this polymerase from this bacteriophage, and it just replicates double and single-stranded DNA regardless of the sequence, okay? That revolutionized Gemini viruses, all right, and the cloning of them. Back when I did it, you had to run these giant prep gels, cut out this band, try to get a tiny amount of this replicative form, figure out what enzyme it cut with, and then it was incredibly tedious. And then this thing came along, and now there's 11 genera here, and there's been 3 more, okay? So now you can't even fit them on one slide, okay? But the big 3 haven't changed, okay? It's begomoviruses, curtoviruses, and mastery viruses. They're the big 3. They're the ones that cause the most disease. They're the ones that have the most members. These other ones have a smaller number of species and in general are not particularly economically important. They just reflect the tremendous diversity of this family of viruses. All right, begomoviruses. Okay, so these are the whitefly transmitted Gemini virus. Okay, and they're transmitted by what we call the whitefly supervector Bemisia tabaci, which exists in a number of cryptic species, which we won't go into here. Okay, and they are, as I mentioned, one of the largest genera of viruses. They have more than 445 recognized ICTV recognized species. And you want a headache? Figure out how you're going to switch that to the new binomial nomenclature, okay? Another topic perhaps we should talk about sometime, okay? And again, they cause a lot of economically important diseases in tropical and subtropical regions. You can see all the crops, including, of course, cucurbits. All right, now, how do these viruses get into plants? Okay, we were briefly talking about. Naturally, they get in by the whitefly. And the whitefly is an excellent vector, okay, including for many of the cucurbit ones, as they've nicely shown for cucurbit leaf crumple virus, Babu and Scott Atkinson. So it's a very nicely whitefly trend. We're going to talk about this issue about seed transmission, but we also need to talk about somebody like me at UC Davis in the middle of agriculture would never be given a permit to work on these viruses if I was using whiteflies, okay? For obvious reasons. If they ever got out, they could. I got permits to work on them only through using techniques that don't involve whiteflies. Mechanical inoculation, as I mentioned, doesn't work with many of them. So it's pretty much agrobacterium delivered or particle bombardment delivered, okay? So we're going to talk about those 2 methods in a minute. Now, begomoviruses have 2 types of genomes. You can have a single DNA, a monopartite genome on the right, which is about 2.8 or 2.9 kb. Tomato yellow leaf curl is a great example of that, okay? And you can see the genes here. I won't talk about those. And then you have the bipartite guys that I already mentioned. They have an A and a B, about 2.6 each, and they're individually encapsulated in virions, okay? They're not together. You have an A virion and a B virion, okay? They're very picky about their size, okay? You try to increase their size, they recombine right back to their normal size. They do not like their genomes being tinkered with in that way, okay? Now, the bipartite begomoviruses are mostly found in the Western Hemisphere. The monopartites are mostly found in the Eastern Hemisphere or Old World, but they've mixed and matched now through the international movement, and as I mentioned, new discovery of monopartite New World indigenous begomoviruses. Some are sap transmissible, the bipartite ones, and we'll mention one of them that is. And examples are bean golden yellow mosaic, bean dwarf mosaic, tomato mottle mosaic, etc. All right. Now, some of them are phloem limited. This is tomato yellow leaf curl. Look at how beautiful that is, huh? That's a capsid protein antibody. Tomato has an inner and outer phloem, and that purple color is how much TYLCV antigen is packed into the phloem of those tomato plants. It's unbelievable. And you can see how it gets up into this is the young developing shoot, and you can see it get right up into the phloem initials. It's wonderfully adapted to the phloem, okay? Okay, but other ones like bean dwarf are not. Okay, they can be mechanically transmissible, and unlike tomato yellow leaf curl, if you mutate the capsid protein of tomato yellow leaf curl, it can't move. Okay, it needs the capsid protein for those virions. But bean dwarf, my baby, I love this virus. This is one of my favorite viruses. And it turns out you can mutate the capsid protein, okay? And you can put something inside it, okay? And that's because the virus is moved by the B component proteins, which have a two-component movement protein system, a nuclear shuttle and a cell-to-cell movement, whereas the capsid protein isn't required. You can mutate it, and the virus moves and causes symptoms, okay? Now, every once in a while, you get lucky. So happens the GFP gene, the green fluorescent protein, which is a fluorescent protein from a jellyfish, was exactly the same size as the capsid protein of BDMV. And so my source shooter, Shauna, postdoc in my lab, swapped the GFP gene for the capsid protein and asked the question, will it make GFP? And sure enough, in a protoplast assay, it makes nice GFP, okay? So now we're like, oh, do we have a reporter that's going to allow us to move our virus around? All right. When I was a kid, I loved this movie, Fantastic Voyage, okay? And it was about shrinking a spaceship down and injecting it into the veins of a guy. And they had to go up and neutralize a blood clot in his brain or something. And he was some famous person or something. Anyway, I don't remember all those details. But I loved how this little spaceship had to navigate. Here you can see it in a blood vessel, okay? How it had to navigate through all the different organs and so on. And of course, if I saw it again today, I'd probably be pretty corny. But when I was a kid, I loved it, okay? And it wasn't because Raquel Welch was the co-star, all right? But it was because this was, to me, really cool, okay? So I wanted to do it with a virus, a plant virus, okay? So we asked the question, would our BDMV tagged virus allow us to follow the virus movement throughout the plant, okay? So here we use the particle bombardment. Here you coat the DNA onto little gold particles and bombard them with helium pressure into these little baby bean seeds, these radicals, and then you plant them, okay? All right, so we have the wild type here, the normal capsid protein. Here's our GFP marker. We inoculate plants. Woohoo! On the left is the MGFP tag virus making symptoms. Here's the wild type. And if you look at and you recognize symptoms, you might say this is a little milder than that. And that's true. The capsid protein does do a little something. Okay. All right. Now, so then we can bombard the hypocotyls and look at the green fluorescence, okay? So the A component with the GFP, it doesn't move, but it makes GFP. Beautiful, just as we know. If you make a mutant in one of the movement proteins, doesn't move, the other doesn't move. The wild type B with the two movement proteins and the A GFP, look at it moving cell to cell. Now we can ask the question, what happens when you bombard these radicals? How deep does it go? Only one cell, okay? Now, we always wanted to know that. How deep does that thing go? Only one cell. And then you can see within 18 hours, it's starting to replicate and make GFP. 24 hours, move cell to cell? Hell yeah. And look at it. It has a direction. It's going right to the phloem. It's not dilly-dallying around. It's going right there. And then if you look like a centimeter above, it's only in the phloem, okay? So now we're following this virus moving in the plant, all right? Now, how about the whole plant? Goes right down to the roots. Roots are very good sources, and many viruses go down to roots as TMV gets tons. Look at this, going up into the phloem, and you know where it's going to. Look at that. Here is the shoot apex, the apical meristem, and grand central station. All of these vascular elements being laid down by protophlome cells, and the virus is attacking them and allowing it to move to all kinds of different places. And using immunolocalization with a capsid protein antibody, we can confirm this protophlome immunolocalization. Look at this going out into the primary leaf. Tons of virus and escaping out of the phloem. But then by 18 days, all of a sudden it's restricted again. And then 36 and 60 days, it's not restricted. And again, here you can see the immunolocalization, a bundle sheath getting into the mesophyll cells. Oh, very nice. Totally consistent with sap transmission. So you can see, depending on the developmental stage of the plant, there's different abilities for the virus to move out of cells, suggesting different levels or different amounts of size for these things to move. Now we get to the flowers. Look at that. Here that's in the flowers, okay? It's moving into the pods. It's in the pod wall. It's in the seed coat. It's not in the funiculus. It's not in the ovary, okay? So in our experiments with this green fluorescently labeled virus, it gets right up into the flowers. It gets into the pods. It gets into the pod walls. These are maternal tissues. It does not get into the fun. So to us, that can lead us to believe it is seed associated or seed borne, but not seed transmitted. And in CIOT, Francisco Morales and his collaborators planted out large numbers of seeds with bean golden, with bean dwarf, and never saw evidence of seed transmission. Now, the other thing to remember about this shoot apex, okay, and the apical meristem, you notice that the virus isn't getting up there, okay? Let's go quickly back here. It isn't getting into that shoot, into that meristem, okay? And why is that? What we know now is gene silencing protects that meristem, okay? And it's not going to let any foreign RNAs or DNAs get into that meristem without a fight, okay? All right. You're probably saying, geez, when's this guy going to start talking about cucurbit begomoviruses, okay? All right, so let's switch to that. So we've got cucurbit begomoviruses that independently emerged in the new and the old worlds, okay? But they're all bipartite, at least as far as I know, okay? They're transmitted by Bemisia tabaci, the super vector, as I mentioned, okay? And they have a propensity to move long distances, okay? As you'll see in one moment, okay? All right, Bill mentioned commonly occur with mixed infections, and that's going to be a problem, as you'll see in one moment. Economic importance varies. Like I told you, cucurbit leaf crumple is not a problem for us anymore, but it is in Florida, and we really need to use IPM to effectively manage them. I hope you can see this okay. So this gives you an example of a few of the key begomoviruses that infect cucurbits, okay? And I'm not going to read through these. Okay, like I mentioned, they're all bipartite. And you can see here, what I want to show here, here's where they originated, but the red is how they've moved to new locations. So if you see cucurbit leaf crumple, southwestern US, but moved to the southeastern US, okay? If you drop down here and you look at squash leaf curl virus, again, emerged in southwestern US, but moved into the Mediterranean basin, where it hooks up with watermelon chlorotic stunt and makes a synergistic infection, okay? And so on, all right? So I'm not going to go through. We're going to focus today on cucurbit leaf crumple and tomato leaf curl New Delhi virus, though, okay? So these are your kind of main cucurbit infecting begomovirus. Now, cucurbit leaf crumple is a very interesting virus. It emerged in Southern California and Southern Arizona and Northern Mexico, okay? And we helped characterize it in 1998. And one of the really interesting things about it that we noticed right off, and I want to shout out to my good friend Tom Tarini, who helped us with this project, okay, and really was the first to recognize this, okay? And it turns out it's a typical, like I mentioned, bipartite New World begomovirus, causes this crumpling and curling, hence the name, okay? It's phloem limited, it requires the capsid protein, but some cucurbits recover from it. So you can see the severe symptoms starting, but when you get away and you see the growth, all of a sudden the symptoms go away. Believe me, we were quite interested in that, okay? So here you can see, we call this recovery, okay? Anytime you see recovery, I don't know if you know what it stands for, rat, okay, you dirty rat, okay? Recovery at top, and recovery at top is associated with gene. So look at how beautiful that is. Hammered by cucurbit leaf crumple, but then recovering. Oh, really cool. That's getting out to the field. You got to do it. Okay. So we developed, Charles Hagan in my lab developed an agro-inoculation system. And again, in this particular case, you put the clone DNAs into a binary vector that you transform into agrobacterium with a disarmed TI plasmid. And then the disarmed TI plasmid will recognize the borders of your binary plasmid and move in what's ever in between your binary plasmid into, theoretically, the genome of the plant in the area that you make this wound with this syringe, okay? And then in about 12 or 14 days, beautiful cucurbit leaf crumple symptoms in the newly emerging leaves. Okay, I love this agro system. It's great for screening. So then we went ahead and looked at some of these cucurbits to see, oh, what about this kind of recovery phenomenon? All right, and look at zucchini, very susceptible. Okay, no matter what, 10 or 20 days, tons of virus. But watermelon, look, by 20 days, you see the amount go down. Okay, but not totally, obviously. And if you look at cantaloupe, though, here's 10 days, look at 20 days. It's way low. Okay, totally consistent with what we're seeing. All right, so again, this is going on in this interaction with cucurbits. It can be bad in the squashes, but moderate recovery in watermelon and very good recovery in cantaloupe. And that's why we don't see this thing as a problem that much in California anymore. Now, interestingly, you can overcome that recovery by co-inoculating with an RNA virus that has a suppressor of gene silencing. So if you co-infect with cucumber mosaic, that recovery doesn't happen. Okay, so that takes advantage of that. All right. So now let's talk a little bit about, are begomoviruses seed transmitted? Okay. All right. So for nearly a decade, we've been getting some of these reports that begomoviruses may be seed transmitted, mostly because you can PCR detect them in flowers and in some of the seed components. Okay. But I want to emphasize here, very importantly, seed-borne does not equal seed transmitted. Okay, just because you detect it in the seed does not mean it's transmitted. And I said with the tomato leaf curl New Delhi, they have done this experiment and they've shown, yes, you can find it in the seeds, but there's no evidence that it's transmitted. Okay, same thing I believe with TYLCV. Okay, it's seed-borne. The evidence that it's seed transmitted is not there. Okay, most of these reports have been in cucurbits and tomato. Okay. A few looked at Benthamiana too. Okay, so again, the other thing is that most of these cases involve agroinoculation, where you're inoculating these young plants. The agrobacterium is transforming phloem cells and dumping the virus into the phloem in the wild type form then, okay, and which is virions, okay? And so you're infecting at an early stage, okay, and giving the virus a better chance, I think, to get up into some of these tissues, okay? So that's needs to be. Now, the other thing, and I really applaud the Spanish group, as I said before, the first report on transmission of TY, it wasn't convincing, okay? There was a number of flaws in it and so forth. And the Spanish did a really thorough grow out, wild type plants infected with white flies, not agro. And I think did a very nice study showing, yes, it's seed borne, but it's not seed transmitted. Okay, they revisited the experiment. If somebody revisits your experiments, you know what they're saying. It means they don't believe you. Okay, all right. Okay, now let's take a look. I hope I can lead you through now some of the results with some of the different viruses. Okay. All right, so this was the first the first kind of seed transmission with TYLCV. It was agro, okay? Do you detect it in the flowers? Yes. Seed, yes. They said they detected it in the embryo. SD means surface disinfestation, removes it from the outside of the seed. I don't think they looked at that. They did say they got it into progeny seedlings, but the seedlings didn't have any symptoms, nor were they able to show that they could take the virus from these progeny and infect the plant and get tomato yellow leaf curl disease. And I never saw them present any evidence of seed transmission in the epidemiology. Okay. Now, this is the Spanish revisiting. They used white flies. They also detected in the flowers and the seeds, but not in the embryo, if I read their paper right. They did show that surface disinfestation removed a lot of it from the outside. So there's a lot of the virus on the outside of the seed because a tomato is loaded with Gemini virus virions, okay? As is the fruits with cucurbit leaf crumple, nicely shown, okay? But again, progeny, they did a grow out test with 3,000 seeds. No, okay? They didn't find TY, they didn't find symptoms, okay? They didn't need to do this. And they also claim there's no evidence of seed transmission in the epidemiological development of the disease in Spain. Okay, again, here's the one with tomato leaf curl New Delhi, agrobacterium inoculation of melon. Okay, yes in the flowers, yes in the seed. Yes, no, I say. It wasn't convincing. It was in the embryo, and they themselves weren't convinced of it. And if you read their paper, they also showed a lot was on the outside that could be removed. They did grow outs, no transmission, and no symptoms in the progeny plants. No, they didn't need to determine this. And I didn't read them saying that they had evidence the thing was coming in for seed. Then let's wrap it. Then we've got cucurbit leaf crumple here, also agro in a very susceptible squash variety, yellow squash, so hypersensitive, okay? Yes, it was detected in the flowers. Yes, it was detected in the seeds. Yes, it was detected in the embryo, quite convincingly in this study. I don't think they did any of the surface disinfestation. They did some limited grow outs, okay, 114. And they did not get wild type symptoms, though they mentioned getting some mild type virus symptoms in some of the seedlings. Okay, they did not do transmission experiments to show this was viable virus. Okay, all right, and also no convincing evidence that seed transmission is playing a role in the epidemiology. And then last was my study I already mentioned on BDMV, okay, using bombardment. Okay, we also detected in the flowers and the seeds, but not in the embryo. And others had shown that it's not seed transmitted epidemiologically. Okay, all right. So I think if you look at this, you can see we're going to be pretty confident that they're seed borne. Okay, we can find it in all of these studies, even in the natural inoculated. Okay, where things get dicey is the transmission to the progeny. And in my opinion, we are not there. As a matter of fact, as I mentioned, the title of this tomato leaf curl New Delhi paper was that the virus is seed borne and not seed transmitted. Okay, so if we look at what the data is telling us now, and we're almost done here. Okay, begomoviruses can be seed-borne in cucurbits and tomato, no question about it. And I think that is not an argument these days. Okay, it's easy. It gets into these maternal tissues very well, moving through the phloem. It's an excellent phloem mover. It's probably virions, but as I told you, the fact that BDMV can move without the capsid protein tells you very clearly that double-strand or single or double-stranded naked DNAs or coded move, non-virion forms, okay? Very important to keep that in mind, okay? But again, most of these cases that have been investigated to see if there's seed transmission using grow-out tests and these type of tests that you really need to do have not supported seed transmission to date, okay? And don't take it from me. Here's a quote from this 2023 paper from the Spanish group saying, okay, epidemiological information available until now, or I put in, still supports the paradigm that transmission of begomoviruses by seed is the exception, not the norm. Okay, so that's probably, you could make it stronger than that at this point, but I think that's a fair statement. Okay. And again, you really need to show that this is a relevant biological thing that's happening in nature. And again, I would say, if it was, why aren't we seeing seeds planted all over in temperate areas and an occasional plant popping up with TYLCV or cucurbit leaf crumple? We don't see that. Okay, as I mentioned, that Cornell finding was traced back to contaminated transplants. Okay. All right, timing is everything. If you're a seed-transmitted virus, like a potivirus, many of them are effective seed transmit. And when you plant an infected potivirus seed, that little seedling shows those symptoms, okay? It's not non-symptomatic in a susceptible host. That's the problem here, okay? So anyway, you see that thing come right up, no question. We do not see that with tomato seeds, with cucurbit seeds, TY showing up in Minnesota or something like that in a single plant here and there, okay? So if this was truly seed transmitted, you would expect to be seeing those types of things. Again, like I say, timing is everything. The agrobacterium inoculation method probably is driving this phenomenon too. We've got to keep all of those things in mind when we're analyzing this data. All right, that's it. Thanks for your attention. I'd like to just thank some people from my lab, Dr. Maria Rojas, Dr. Mysore Sudarshana, who made a lot of these contra. Han Li Wang was really good at doing the plant dissection and so forth to follow that, to have that fantastic voyage. Dr. Charles Hagan, who made the agro system for cucurbit leaf crumple and then did the recovery work, and my longtime collaborator, Bill Lucas, on the plant stuff. Bill, my longtime collaborator on cucurbit virus, the great Tom Turini, farm advisor in Fresno County, who we all rely on, our eyes and ears out in the field these days, and he's happy that he's not seeing tons of viruses in cucurbits this year, I'm sure. So anyway, thanks again.